In the HL section, for A7 chromatography, we are required to describe the techniques of gas-liquid chromatography, GLC, and high-performance liquid chromatography, and deduce which chromatographic technique is most appropriate for separating the components in a particular mixture. High-performance liquid chromatography, HPLC. HPLC is a form of adsorption chromatography with an aim of separating mixtures containing non-volatile substances either to analyze the mixture or to separate a required product in a reaction mixture. The mobile phase is a high purity solvent while the stationary phase is contained in a HPLC column that consists of uniform porous silica particles with surface pores that are extremely small to increase surface area. The mobile phase is forced through the column using extremely high pressure, which results in better separation relative to liquid chromatography. These separated components is usually detected by ultraviolet radiation as they pass through a flow cell. The output recorded is on a piece of chromatogram. In this case, the chromatogram would have absorbance on the y-axis and time on the x-axis. The next slide represents a schematic diagram of HPLC. As you can see, we have a huge mobile phase reservoir that enters the high pressure pump via capillary action. The high pressure pump provides the necessary drive for the mobile phase to pass through the column and allow the different components in the particular mixture to be separated. The separated components of the mixture will then pass through the flow cell where it is being detected using ultraviolet radiation, and this is then plotted on a chromatogram of absorbance against time. This particular slide, it shows a diagram of a column, and inside this column, the sample, which contains 3 components in a particular mixture, is now going to be separated using this particular technique. Now, as you can see from this mixture, you would realize that the 3 different components have differing levels of affinity for the mobile phase. The one right at the bottom has the highest affinity for the mobile phase, and it tends to elute out first. And this is followed by the cubes and then the sun. And it is then detected on a chromatogram, as illustrated here, of signal against time, where each particular peak is characteristic of a particular component in the mixture. The time taken or the time lapse for when the sample is injected to when a particular component elutes out is known as a retention time. And each retention time is characteristic of a particular component for the same column and temperature. The area under each peak indicates the relative quantities of each component that is present in the mixture injected. In this particular diagram, glucose also have the shortest retention time, meaning it has the greatest affinity for the mobile phase. The main advantage of HPLC over LC, liquid chromatography, is that the analyte is forced through the column by the mobile phase at such high pressures that the time that separated components remain on the stationary phase is decreased, and this will result in narrower peaks. And narrower peaks will result in a better separation. Section to be covered is gas chromatography. And according to our learning objectives stated earlier, we are required to describe its techniques. Gas liquid chromatography, or GLC, is a form of partition chromatography with an aim of separating mixtures containing fairly volatile substances, either to analyze them or to separate a required product in a reaction mixture. The mobile phase is a gas that carries the components of the mixture over the stationary phase. The stationary phase is packed in a tube called a column that is coated with a high boiling point liquid. Injected sample is first vaporized and carried by a carrier gas, commonly nitrogen and helium, that acts as the mobile phase. The mobile phase then passes into the column over the stationary phase, and the components in this mixture would then partition between the mobile and the stationary phase. At the end of the column, there is a detector, and this detector, commonly known as a mass spectrometer, detects analytes that are unknown. For analytes that are known, we usually use a flame ionization detector. However, the name of this detector is not required in your syllabus. This is a schematic diagram of GLC. At the start here, we have a tank that contains the carrier gas. The sample is first injected through an injection port using a hypodermic needle and is then vaporized due to the temperature in the heated chamber. The carrier gas from the tank then transports it into the column, and as it passes into the column, it partitions itself between the mobile phase and the stationary phase. At the end of the column, a detector detects each component of the mixture as it elutes out of the component, as well as measuring its concentration. As said earlier, the area under the peak is proportional to the concentration of each component of the mixture. As you can see here, there are a total of 2 components in the analyte. This particular slide depicts a portion of the column, and as you can see represented by the gray arrow here, it kinds of represents the hypodermic needle. As it injects the sample into the injection port, the sample is first vaporized, and because the sample contains components that have different levels of affinity or volatility, it causes them to separate out between the mobile and the stationary phase. As you can see here, the sample is made up of 2 components, the yellow and the blue dye. And as it passes through the column into the detector, represented by the gray box, the detector then plots signal recording against time. And as you can see here, the first peak represents that of the yellow dye, while the second peak represents that of the blue dye. The area under each peak is proportional to the relative concentration of each particular dye, and as you can see on this chromatogram, the 2 dyes have an almost equal relative concentration. One could also observe from the chromatogram that there is proper separation between the 2 dyes, judging from the gap between the 2 peaks. In this slide, it shows the GLC being linked to a mass spectrometer. Now, why is the purpose of doing so? Now, let's say, for example, you have an unknown mixture, and this unknown mixture contains a certain number of components, and the role of the GLC, obviously, is to separate the components out. However, the GLC cannot discern its identity. We require an instrument that is able to do so. And because of that, we use a mass spectrometer to discern the identity of the components in the mixture.